Bismillahirrahmanirrahim. Assalamu alaikum and welcome to Biosciences with Shatreen Sawati. And today I'm here with my lecture from coronavirus series, and this is almost fifth lecture from that series, and this will be about the SARS coronavirus vaccine, especially messenger RNA vaccine. And before starting my lecture, I have a humble request to all of you. If you did not subscribe my channel, kindly subscribe my channel and promote my channel, like my videos, share my videos. If you're sharing my videos, you are sharing the knowledge. So keep watching and keep sharing knowledge with other. And kindly do promote and do viral the educational content and knowledge. Okay, now I'm going to start my lecture about the SARS coronavirus messenger RNA vaccine. This is very interesting topic, and I created this lecture on the basis of all the research articles available, and I'm very thankful to New York Times. I got a lot from New York Times, and also I'm thankful to the Pfizer and BioNTech companies. I got a lot of knowledge from their website, so I created this lecture. So must watch this lecture. uh, most of the people around the world are so much worried and conscious about the SARS coronavirus vaccine, whether it is available, not available, will be effective, not effective, and many more concerns. So, in this lecture, I will discuss about the first of all, I will give you the introduction, then new approach to the messenger RNA vaccine and advantages of the messenger RNA vaccines, and how do messenger RNA vaccine work, and which vaccines are approved by the FDA and now available in the market. I will discuss all these things step by step. So, how do we control the coronavirus? We can control the coronavirus by adopting the following prevention measures like wearing mask, washing hand, and also must keep the distance of about 6 feet. And the last one is the vaccine, which I'm now going to discuss. Especially, this topic is about the messenger RNA vaccine. Vaccines typically require years of research and testing before reaching the clinic, but in 2020, scientists embarked on a race to produce safe and effective coronavirus vaccine in record time. Researchers are currently testing 68 vaccines in clinical trial on human, and 20 have reached the final stages of testing, and maybe more. Um, it, at least 90 preclinical vaccines are under active investigation in animals. Now, the new approach to vaccines. First of all, we should know that what is messenger RNA and where it produced, and what is the function of the messenger RNA. So, messenger RNA produce are formed during transcription from DNA in the nucleus, and then this transcription, this messenger RNA then move to the cytoplasm and do translation and make the proteins. And especially in this case, messenger RNA vaccine for SARS coronavirus, scientists created the hybrid messenger RNA, and that messenger RNA is responsible for the formation of the spike protein. So, must watch my lecture about the spike protein. You will get a lot, and this lecture will be easily understandable for you people. So, messenger RNA vaccines are a new type of vaccines to produce or to protect against the infectious diseases and to trigger an immune response. Messenger RNA vaccines teach our cells how to make a protein or even just a piece of the protein that triggers an immune response inside our body. Immune immune response produce antibodies that protect us from getting infected if the real virus enter our body. Is it clear? So, messenger RNA vaccine have been studied before for flu, Zika, rabies, and cytomegalovirus. Successfully, these vaccines are working. As soon as the necessary instruction about the virus that causes COVID-19 was available, scientists began designing the messenger RNA instructions for cells to build the unique spike proteins into an messenger RNA vaccine. So must watch my lecture about the spike protein. You will understand the function of the spike proteins in infection. So that's why the scientists are now creating the messenger RNA, which is responsible for producing the spike proteins. Messenger RNA vaccines are also called mRNA vaccines, are some of the first COVID-19 vaccine authorized for use in United States. Advantages of messenger RNA. Now, what are the advantages of them? Because most of the people have a concern with the advantages, with the side effects, and many more. So, messenger RNAs are non-infectious, and clinical trials have shown no risk of infection or insertional mutagenesis. Because this hybrid messenger RNA, which is messenger RNA vaccine, when administered into the body, then this messenger RNA do not enter into the human cell nucleus and do not damage the DNA or cause any mutation. In other words, there is no risk of creation of mutation of DNA due to artificial messenger RNA. Messenger RNA is degraded and excreted by natural normal cellular processes. So it is safe to use. Messenger RNA vaccines have shown safe outcomes in animals and are adaptable for rapid application to new infectious diseases. Additionally, the short manufacturing time, this is very important and very important advantage for controlling this rapid and pandemic coronavirus. So the short manufacturing time inherent in messenger RNA production is advantageous to avoid contamination with other viruses, a concern presented in the production of other vaccines. Is it clear? So, how do messenger RNA work? This is very important. Now, I will tell you how. As I have discussed in detail about the structure and genome and the spike protein in my previous lecture, so must watch all these lectures. Then this lecture will be easily understandable for you people. So this is the structure of the coronavirus, and these are the spike proteins present on the surface of the coronavirus, and these spike proteins help in infection. These spike protein help in the fusion of the coronavirus with the human cell and then causes the infection. So inside the coronavirus, the genome of that coronavirus is single stranded RNA, positive sense RNA, which act as a messenger RNA. And this genome consists of the 6 open reading frame, and one of the most important open reading frame is the structural gene that is responsible for the production of the spike protein, S proteins. So this region, just suppose that the spike producing protein producing region, which is the S open reading frame, and scientists created this from this portion, from this gene, the messenger RNA that is responsible for the production of the spike protein. Now, this messenger RNA is a hybrid type of messenger RNA synthesized in the lab, and I will discuss in detail about the hybrid messenger RNA. So here I'm not going to discuss about the hybrid type of messenger RNA. So this hybrid messenger RNA then what will happen? Surrounded by the lipid nanoparticles. Why the scientists wrap this messenger RNA in the lipid nanoparticles, I will discuss later. So this messenger RNA, which is wrapped by the lipid nanoparticle, when administered into the body of the human, then what will happen? This messenger RNA will move to the cell and then enter into the cell, and what will this messenger RNA will do, I will discuss later. So messenger RNA inside an oily shell means the lipid nanoparticles surrounding the messenger RNA. The vaccines uses messenger RNA, genetic material that our cells read to make proteins. Our cells, human cells, read this messenger RNA and make the proteins, make the spike proteins. So the molecules called messenger RNA for short is fragile and would be chopped, means destroyed by the enzymes, naturally present enzymes, naturally occurring enzyme inside the cell of the human body. So if it were injected directly into the body, it will be destroyed. So that's why what will happen, the scientists wrapped this messenger RNA in the lipid nanoparticle. That's why it is surrounded by lipid nanoparticle. So the concept clear about that. So to protect the vaccine, wrap the messenger in the oily bubbles made of lipid nanoparticles. The question and the answer is clear upon you people. Now I'm going towards the next. Entering a cell, what will happen? When the drug administered into the body, must read this statement. This is the messenger RNA vaccine, which is wrapped by or surrounding by the lipid nanoparticles. When this vaccine messenger RNA enter into the cell, then what will happen? This messenger RNA move towards the ribosomes of the human cell. Then this messenger RNA, what will happen? Undergo the translation process and produces the spike proteins. Means this messenger RNA carry the instruction for the formation of the spike protein. So what will happen? Spike proteins produced by translation inside the human cell, as you can see here. And after, what will happen? After undergoing the mechanism of translation and producing the spike protein, this RNA, messenger RNA, degraded by natural process. Is it clear? And you can see and you can read this statement about that. Now, I'm moving towards the next slide. Some of the spike proteins form spikes that migrate to the surface of the cell. Now, after producing these spike proteins, then migrate to the surface of the cell. As you can see here, these move outside surface of the cell and stick out their tips. The vaccinated cells also break up some of the proteins into fragments. As you can see here, spike proteins also break down and into fragments, which they present on their surface. You can see here this vaccinated cells presented these spike proteins on its surface. These protruding spikes and spike protein fragments can then be recognized by the immune system. Now, what will happen? The next story will start. The next process means the immune system now become active when the spike proteins produced inside the vaccinated cells when move towards the outer surface, and then this cell vaccinated cell is detected by the immune system. Now what will happen? Spotting the intruders. When a vaccinated cell dies, then what will happen? When the vaccinated cell die, the debris will contain many spike proteins and protein fragments. Is it clear? Which can then be taken up by a type of the immune cells called an antigen presenting cell. As you can see here, this is the antigen presenting cell. What will happen? When this cell die, when this cell die, the debris of that cells contain the spike proteins and fragments of the spike protein. Then what will happen? There is a cell which is known as, which is the most important part of the immune system, is the antigen presenting cell. This antigen presenting cell, then what will happen? Eat the dead debris and presenting the spike proteins on its surface. Then what will happen? As you can see here, there is another type of the cell which is known as the T cell. T helper cell. Now what will happen? The cells present fragments of the spike protein on its surface. When other cells called the helper T cell detect these fragments, the helper T cells can raise the alarm and help marshals other immune cells to fight the infection. Now, as you can see here, there are the helper T cells. These helper T cells then activate the B cells. And B cells are actually those cells which produces the antibodies. Now, our human immune system detected the spike proteins which are produced by the messenger RNA vaccine. Then after detection, what will happen? The B cells produces antibodies against that spike proteins. Is it clear now? Now what will happen? After the production of the antibodies, as you can see here, antibodies are produced by the B cells. When the helper T cells activate this B cells produces the antibodies against the spike proteins. Now, other immune cells called B cells may bumps into the, you can also read this statement, bump into the coronavirus spikes on the surface of the vaccinated cells are free floating spike protein fragments. A few of the B cells may be able to lock onto the spike proteins, as you can see here. If these B cells are then activated by the helper T cells, they will start to proliferate and pour out antibodies that target the spike protein. As I have discussed, that what will happen, as you can see, this is the antigen presenting cell. This antigen presenting cell is now detected by the T cells, helper T cells. Now this helper T cells instruct the B cells to produce the antibodies, and these antibodies which are produced against the spike proteins, now what will happen? Fight the produces these antibodies, fight the infection later on when the real virus enters into our body. Okay. Now, other immune cells, as you can see here, which are the B cells, which produces the antibodies, what will happen? The antibodies can latch onto the coronavirus spike, mark the virus for destruction, and prevent infection by blocking the spike from attaching to the other cells. Is it clear? Whenever the real virus, which is the coronavirus, after when the person is vaccinated, now the person is vaccinated and antibodies have been produced inside the bodies, then what will happen? when the antibodies have been produced inside the body, when the coronavirus enter, these antibodies detect the spike proteins and come and attach with these spike proteins of the real virus, which is the coronavirus. Now, what will happen? This spike protein is unable to attach or fuse with the human cells. And in this way, the human will be prevented by the coronavirus. And when these antibodies attach with the spike protein and then block and destroy them, spikes. Okay. Now what will happen? Killing infected cells. The antigen presenting cells can also activate other type of the immune cells, which are the T helper cells, to seek out and destroy any coronavirus infected cells that display the spike protein fragments on their surface. As well as the B cells are activated by the helper T cells, as we know that. Now what will happen? These antigen presenting cells also activate the killer cells. And what will happen? These killer cells also destroy the coronavirus infected cells. Is it clear? On one side, antibodies have been produced in the body as a result of the messenger RNA vaccine. And on the other hand, what will happen? The antigen present cell also activate the killer T cells, and this killer T cell, then what will happen? Take the coronavirus infected cell and destroy them. Now, available messenger RNA vaccines. I will show you which vaccines are now available. The two COVID-19 vaccine candidates, one from the Pfizer BioNTech and the other from the Moderna, have received emergency use authorization from the Food and Drug Administration for the first time outside of the clinical trial environment. Vaccination begins in the United States in mid-December 2020. So, which two vaccines? One is the Pfizer and the other is the Moderna. Both are authorized from the Food and Drug Administration for the first time use. Okay. Now, how effective is the coronavirus COVID-19 messenger RNA vaccine? The Pfizer and Moderna vaccines perform similarly. Their efficacy, the Pfizer vaccine is 95% effective. Its efficacy is 95%, and Moderna vaccines 94.1% effectiveness. And this vaccine administered intramuscularly. Now, remembering the virus. How do the vaccine work? The Pfizer BioNTech vaccine required 2 injections given 21 days apart to prime the immune system well enough to fight off the coronavirus. But because the vaccine is so new, researchers don't know how long its protection might last. Okay. Preparation and injection. You can see each vial of this is about the Pfizer and Moderna. There's 5 doses of 0.3 milliliter, and vaccines must be thawed before injection and more. So this is about the preparation and injection. You must read. Okay, storage. This is about the storage. Because of their fragility, and the messenger RNA molecule will quickly fall apart at room temperature. Pfizer is building a special container with dry ice, thermal sensors, and GPS trackers to ensure the vaccines can be transported at 94 degrees centigrade to stay viable. So this is very important for the storage of the vaccine. Especially I'm talking about the messenger RNA vaccine. Okay, many of the people ask that if I already had COVID-19, should I get the vaccine? Now, the answer is it is recommended to get the vaccine if even if you have had COVID 19 because it is unknown how long the body's protective mechanism will last. It is suggested that the artificial vaccine may provide a more robust immune response than being infected with the virus naturally. I hope so. The lecture will be clear upon you people. So if you have any question, write in the comment box. And thank you very much. And thank you to all. I got this information from many websites and from used lots of research articles. So I'm thankful to all of them. And thank you very much, and I will be back with new lecture inshallah soon. Till then, Allah Hafiz.